Professor, Physician, Epidemiology. I am Dr. Sri Banerjee. Hello everyone, I'm Dr. Sri Banerjee, core faculty for the College of Health Sciences and Public Policy at Walden University. In this video, what I wanted to do is take some bench science research and kind of connect it to clinical sciences, medical sciences, and the area of population/public health. So let's get started here. Why are we interested in even thinking about pancreatic beta cell-like development? Is because pancreatic beta cells are, in fact, the cells that are through either autoimmune mechanism or some other mechanism actually is affected. So that's the reason that this article is being studied. So thinking about, so that's the public health perspective, diabetes. So thinking about, I already said I was going to connect this to those of you who do laboratory work. This is looking at specifically at stem cells and different types of stem cells that may have been effective in at least influencing diabetes. And diabetes is a metabolic disease, as you know. It not only affects glucose metabolism, but also lipid and protein metabolism. So we already know that. Stem cell therapy is an efficient method for the treatment of diabetes, which can be achieved by differentiating pancreatic beta-like cells. So that's the idea. And I actually have worked with stem cells myself, injecting stereotactically injecting stem cells into the brain of a rat in order to study movement disorders such as Parkinson's disease. But these were not embryonic stem cells. These were more totipotent stem cells. The types of stem cells and how they are being used in research, this is getting more and more important. If you're interested to find out more about some of these smaller molecules that they talk about in terms of stem cells and Wharton's jelly from the umbilical cord and umbilical stem cells, then you can kind of go into this. This goes into a lot of detail as to the types of endocrine markers, pancreatic endocrine markers that can come out of differentiated stem cells. So this is a robust area of research. And then what I would add here is to check out this different place where there is kind of a distinction of the different phases of the different trials. And let me show the connection to where you can check out more information about each of these. But before this, let me go ahead and widen this table for you so that you can see the whole thing. So on the left-hand side is the pathological condition, autologous mesenchymal stromal cell. 24 patients were enrolled. So this was not a massive study. This was a small study. But the one that was the smallest study was the diabetic nephropathy with 15 only enrolled patients. So human umbilical cord mesenchymal stem cells was the intervention. And the outcome measures that was measured was incidence of treatment emergent, treatment chronic adverse events, and estimated glomerular filtration rate. to try to see if these umbilical cord stem cells actually made a difference in trying to in trying to address nephropathy, kidney damage that comes about from diabetes. So since we're looking at that one, let's take a look at the intervention type. So let's switch screens here. So now what I did here is I switched over the NCT number that you saw in the other paper because I wanted to know more about this small n intervention study. I'm actually currently working on a smaller n intervention study, and so this is something that can provide insight that on that as well. So here's the specific study. And this is coming out of Yan'an Hospital of Kunming City, China. And the intervention is human umbilical cord mesenchymal stem cells. Again, they're still recruiting for this. Let me switch the screen over a little bit so that you can see this a little bit better. But this is from clinicaltrials.gov. Then now you can find out exactly where they stand with the phase of their study. No results have been posted yet, but here are the details. This is very early phase 1 clinical trial data analysis. The number of adverse events and the number of severity adverse events were determined based on the changes in the data summary. So some descriptive statistics is probably the best that they can provide. Any sort of relational data will need to be, and significance testing may need more power to be able to complete. So this is an example of how you can take a clinical trial, or in this case, actually, several clinical trials, and kind of look it up a little bit more and see what the details are. As I had said, the primary outcome measure is the patient's discomfort and clinical symptoms during the study that were recorded. That was there, but the primary outcome, of course, was the incidence of treatment emergent and treatment chronic adverse events that were taking place. So any sort of basically physical examination abnormalities, those were recorded. Lab test indicators, if those came about with this treatment with umbilical cord stem cells, those were recorded. So stem cells are a frontier land still. Actually, I worked on the research that I talked to you about 20 years ago, but it's still a frontier land. More is being explored and found. Here's the enrollment criteria if you want to sign up. And so here's the exclusion criteria as well. And each one of these are set up in the same way. So you can take a look at all of this and see, if you do actually qualify for this. And so you can contact, here's the email that you can contact. You can also call them if you want to find out more information. And here's the responsible party, the hospital, and clinicaltrials.gov indicator. All the history of changes are recorded, and everything is all documented because this is FDA. This is one of the clinical trials. So circling back to the paper, it's still all some of this similar things. Stem cell research is something that is still being explored in the context of chronic diseases and just as a potential intervention. So look at this as a topic sort of recording, content, and methodological recording. Thank you for listening.